Hello students, welcome to SK Biology. Today we are going to study transcription in prokaryotes. Transcription is the process by which genetic information present in double-stranded DNA is copied into RNA by enzyme RNA polymerase. There are some key features of prokaryotic transcription process. The site of transcription in bacteria is cytoplasm, as DNA is not enclosed in nucleus. Second feature is only one enzyme, which is DNA dependent RNA polymerase, can synthesize all type of RNA in prokaryotes. No any post-transcriptional modification is required in prokaryotes. That is, after production of RNA, it is not modified. Next feature is in prokaryotes, transcription, that is DNA to RNA conversion, and translation, that is RNA to protein conversion, is coupled. That is, it occurs simultaneously. Let us understand enzyme involved in transcription process. RNA polymerase, important in prokaryote, is also called as DNA dependent RNA polymerase, as it uses DNA as a template to synthesize RNA. This is the structure of enzyme containing 6 subunits. Core enzyme contains 2 alpha subunit which functions for enzyme assembly and promoter interaction. Second subunit is beta subunit required for RNA synthesis. Third subunit is beta prime which is required for DNA binding. Another subunit is omega, it required for enzyme stability. The sixth subunit is omega factor which is responsible for promoter recognition. DNA polymerase without sigma factor is called as core enzyme, and when sigma factor is added to core enzyme, it became holo enzyme. Sigma factor is very important as it helps RNA polymerase to recognize promoter region. Now let us understand promoter region in prokaryotes. It is the sequence of DNA where actually RNA polymerase binds. Promoter region contains 2 important sequences. Minus 35 region, which is having sequence TTGA-CA, and minus 10 region, which is also called as Pribnow box. The sequence present is TATA-AT. It is also called as Tata box. Sigma factor recognizes these 2 sequences to start transcription, and RNA polymerase binds at promoter. Once transcription unit and RNA polymerase, that is holo enzyme, come together, prokaryotic transcription occurs in 3 steps: initiation, elongation, and termination. Let us understand these steps one by one. So, first step is initiation. Here, RNA polymerase along with sigma factor bind to promoter region and unwinds DNA, forming small opening. We call it as a transcription bubble. RNA synthesis starts at +1 site. After synthesis of nearly 10 nucleotides, sigma factor detaches and core enzyme continues transcription. Sigma factor is required for initiation only. Second step of transcription in prokaryote is elongation, where RNA polymerase core enzyme moves along the template DNA strand in 3' to 5' direction, and RNA is synthesized in 5' to 3' direction. Complementary pairing occurs in nitrogenous bases. A present in DNA template is added as a U in RNA synthesized. In the same way, T present will be paired with A, G will be paired with C, and C will pair with G. As RNA contains uracil instead of thymine, the adenine present in DNA template will be added as a uracil in synthesized RNA. This process does not require any primer. In this way, complete elongation will happen. Now, the third step of transcription, which is termination of transcription, so that the transcription will come to an end. There are 2 types of termination in prokaryote. First one is Rho dependent termination, in which Rho protein binds to transcription machinery at root site and separate newly synthesized RNA along with DNA template and RNA polymerase. Another type of termination is row independent termination, in which row protein is not required. Instead of row protein, the newly synthesized RNA forms loop structure at GC-rich region, followed by stretch of uracil residue. AU is weak bond as it contains only 2 hydrogen bond, so it can detach RNA easily and stops termination process. So this is all about transcription in prokaryotes.